Okay, I would like to refresh back what we have already learned so far. So previously we have learned about glycolysis, okay, where the glucose is being broken down into pyruvate, and then if there is oxygen, so this is an aerobic respiration, so the pyruvate will be further oxidized and enters the mitochondrial matrix and proceed with link reaction. And then after that, during the link reaction, the pyruvate converted into acetyl-CoA, and then acetyl-CoA enters the Krebs cycle, fits the acetyl group into the Krebs cycles, and will be further oxidized. And throughout the process of glycolysis, link reactions, and Krebs cycle, we produce the NADH as the electron carrier and also the FADH2. So if you can see in this diagram here, during glycolysis, we already have produced 2 molecules of ATP, net 2 ATP, and for the Krebs cycle also, we already produce 2 net ATP. So altogether, we already produce 4 ATP during glycolysis and also Krebs cycle via substrate level phosphorylation. Okay, so where the rest of the ATP comes from? So the rest of the ATP actually comes from the process we call oxidative phosphorylation. Okay, so if you can see here, the NADH here and also the FADH2 here, they are the electron carrier. They will bring all these electrons to the electron transport chain, and in the electron transport chain, together with the process called chemiosmosis, together these two processes will produce more ATP. It's either 32 ATP or 34 ATP. This ATP is what we say produced in oxidative phosphorylation. So today we are going to learn in details how oxidative phosphorylation occurs. Okay. So oxidative phosphorylation is ATP formed using energy derived from the redox reactions of an electron transport chain. Okay. Oxidative here is referring to the redox reaction. If you guys remember, redox reaction involving process of oxidation and reduction. Oxidation means the molecule give out or donates its electron. Reduction means the molecule accept the electrons. Okay, and this happens at the electron transport chain. Electron transport chain is the complexes of protein or electron carriers embedded in the inner membrane of the mitochondria. This is the mitochondria. So mitochondria got 2 membrane, inner membrane and outer membrane. So the electron transport chain location is embedded in the inner membrane of the mitochondria. So this is one of the, you see in the diagram here, this is one of the electron transport chain shown in the inner membrane of mitochondria. Okay, so in order for the ATP to be produced, this electron transport through the electron transport chain will be coupled with the process of chemiosmosis, and at the end of the day, through the oxidative phosphorylation, we will got the ATP here. So you can see in this diagram, at the end of the day, ATP is being produced here. Okay. Okay. So we have got 4 ATP out of 38 ATP produced by respiration of glucose through the substrate level phosphorylation that we have discussed, happening in the glycolysis and also Krebs cycle. So most of the ATP comes from the energy in the electrons carried by the NADH and also FADH2, where NADH is produced in glycolysis, in link reactions, in Krebs cycle, NADH is produced. And where FADH2 is produced, FADH2 is produced in the Krebs cycle, isn't it? So these 2 electron carriers, they will carry the electrons, and they will carry the electrons to the inner membrane of mitochondria and pass the electrons to the electron transport chain that is embedded in the inner membrane of the mitochondria. Okay, so the energy in these electrons is used in the electron transport system to power the ATP synthesis. Okay, so these electrons carried by the NADH and FADH2, they have energy, and once they pass the energy to the electron carrier of the electron transport chain, energy will be released from the process, and that energy will be then subsequently used to power the ATP synthesis, and at the end of the day, ATP will be produced. ATP will be synthesized. How many ATP? It's either 32 ATP or 34 ATP. So these all happening in the oxidative phosphorylation. So let's know what is electron transport chain. Electron transport chain is a collection of molecules. Okay, it's a collection of molecules serve as electron carriers embedded in where? In the inner membrane of the mitochondria. So this inner membrane, if you still remember, is they are they are folded, or the infolding of the inner membrane is also known as the cristae. Okay, cristae. So this cristae, this inner membrane known as the cristae is folded to increase its surface area and provide space for thousands of copies of the chain in each mitochondria. So you guys see this. So this is the inner membrane of mitochondria. So this is the plasma membrane, isn't it? This is the plasma membrane. So this purple color, this is the protein complexes embedded in the inner membrane. So there are thousands of copies of this electron transport chain in the inner membrane mitochondria. In this diagram, it only shows you one of the chain, but actually, in reality, in the membrane, there are thousands of this electron transport chain. And you see here, this is the matrix. Okay, mitochondrial matrix here. This is the membrane, and this is the intermembrane space. Intermembrane space, ruangan antara membrane. Antara membrane, which membrane? The outer membrane. The outer membrane, and also this is the inner membrane. Okay. So most components of the chain are proteins, which exist in multi-protein complexes numbered 1 through 4. So you see this one, this is protein complex 1 is number 1 here. So this is protein complex 1, and then this is protein complex 2, and this one is the protein complex 3, and this one is the protein complex 4. Okay. So altogether in one electron transport chain system, there are 4 protein complexes, number 1, 2, 3, and 4. Okay, but actually these protein complex, they have their own specific name, which we will learn later. But to simply, to easily say, we just numbered it as 1, 2, 3, and 4. Okay. And then tightly bound to these proteins are prostatic group. If you still remember, prostatic group, you guys learned in the chapter of enzyme, where prostatic groups are the cofactor of enzyme. Okay, cofactor of enzyme. They are the non-protein components, okay, which accept and also donate electrons. So all these prostatic group, they are bounded to all these protein complexes to help accept and donate electrons. Okay, to help accept and also donate electrons from one carrier to another. Okay. So what happened during the electron is being transferred, being transported along the chain. So during electron transport along the chain, electron carriers will alternate between reduced as it picks up the electron and oxidized as it give up electrons. Okay, so electron transport chain is the rantaian, is the chain where all these electron carrier, they will accept electrons. They will accept electrons. Once they accept the electrons, they become reduced. And once they accept, then they pass to the next neighbor, pass to the next electron carrier. Once they pass the electrons to the next carrier, to the next neighbor, they become oxidized. And the next neighbor that accept the electrons then become reduced once it accept the electrons. And the process will be repeated. And the neighbor, once it accept the electron and being reduced, next, it will then pass to the next neighbor, and the next neighbor accept the electrons and become reduced. Okay, and the one that giving out the electrons become reduced. Okay, so become oxidized. Okay, so this is what we see redox reaction. That's alternating oxidation process and reduction process occurring once the electron carrier accept and donates the electrons. So each component of the chain accept electron from its uphill neighbor. Okay, the one that give the electron is from the uphill, from the, we call it uphill neighbor, which is less electronegative. Okay, and then pass the electron to its downhill neighbor, which is more electron negative neighbor. So if you see here, complex 1, complex 2, complex 3, complex 4, if I ask you which complex is more electronegative, electronegative means more tendency to accept the electrons, more tendency to accept the electrons. That's why it's more electronegative. So if complex 1, 2, 3, and 4, which one is more electronegative? Anyone can answer?
2 and 3.
No, complex 1, 2, 3, and 4, which protein complex is the most electronegative? 1, 2, 3, or 4?
4.
4. Yeah, 4. So yeah, the answer is 4. Okay, so complex 1 will at the end passes the electrons to complex straight to complex 3. Okay, complex 1 will pass the electrons to coenzyme Q, and then coenzyme Q pass the electrons to protein complex 3, and then pass to cytochrome C, and then pass to complex 4. So if I ask you which protein complexes is most more electronegative, that means the complex 4 lah. Okay, the one that pass out the electrons is the uphill neighbor. They will pass the electrons to the downhill neighbor. The downhill neighbor, which is the next neighbor, is more electronegative, okay, than the before one. Okay, so in aerobic respiration, the final electron acceptor of the chain is oxygen. Okay, so this electron will be continually passed from one carrier to another. But what is the final electron acceptor? The last acceptor that will receive the electron. So the answer is oxygen. So you see here in this diagram, this is oxygen. So oxygen, if the question asks you what is the function of oxygen in the electron transport chain, the answer is as the final electron acceptor. So this oxygen will accept the electrons together with protons and will form water molecule. So that's why we are continuously need to breathe in oxygen in order for the aerobic respiration to occur, because oxygen is the final electron acceptor. If there is no oxygen, that means we don't have the final electron acceptor. So this process of electron transport will be disrupted, and the cellular respiration on the whole will be disrupted, affected. Okay. So as electron pass through the chain, a series of exergonic reactions will release small amounts of energy. So as the electrons pass from one carrier to another, the energy will be released. Energy will be released from the electrons. So the overall energy drop for electrons traveling from the NADH and NADH2 to oxygen, which is the final electron receptor, is broken up into a series of smaller steps by the ETC. Okay, so this diagram shows the overall in detail the protein complexes in one electron transport chain. You can see here, this is complex protein number 1, and then this is the complex protein number 2, and then this is the mobile electron carrier coenzyme Q, and then this is protein complex number 3, which having the cytochrome B, ion sulfur center, and cytochrome C1 in it. And then we have the cytochrome C here as the mobile electron carrier. And the last one, we have the protein complex 4, which consists of the cytochrome A and cytochrome E3. You can see the arrow here. What is this arrow show? So this arrow actually shows the electron is being transferred from one molecule to another. Where actually the electrons come from? So the electrons come from this one. You see here NADH and the FADH2. So these are the electron carrier that pass the electrons to all these protein complexes. Again, where does this NADH and where does this FADH2 come from? So as what we have learned previously, NADH and NADH2, they are produced from the process of glycolysis. They are produced, NADH is produced from the process of glycolysis and lineations and Krebs cycle, isn't it? And also FADH2 is being produced from the process of Krebs cycle. So once they are produced there, so they will go to the inner membrane of the mitochondria, and then they will pass the electrons to all these protein complexes embedded in the electron transport chain. And then in this electron transport chain, you can see the arrow here. The arrow here shows electron is being transported from one electron carrier to another. And at the end of the day, where the electrons go, where the electron is being accepted, the electron is being accepted by who? By which molecule? So you can see at the end of the day here, the electron is being accepted by the final electron receptor, which is the oxygen. Okay, and the oxygen will accept the electrons together with protons here in the surrounding, and it will become water. Okay, the product will become water at the end of the electron transport chain reactions. Okay, so now let's take a look in detail one by one. We are going to see what happened in each of the protein. Okay, before that, complex 1 here, complex 1, also known as the NADH dehydrogenase. This is the name for the complex 1. If the question asks you name complex 1, so the answer is NADH dehydrogenase. Okay, this complex protein NADH dehydrogenase, they contains the FMN and also iron sulfur protein. Okay, next one, we have the complex 2. This is complex 2. Okay, you can see here, complex 2 here contains the FAD and iron sulfur center. Complex 2 also known as succinate dehydrogenase. If the question asks you what is the name of complex 2, the answer is succinate dehydrogenase, or easily said complex 2. Okay, and then we have the complex 3. This is complex 3. Complex 3 also known as the cytochrome C reductase. And it contains cytochromes. To be specific, you can see in this diagram, cytochrome B here and also cytochrome C1 here. And also they also got one iron sulfur center or iron sulfur protein. Okay. Next one, we have complex 4. This is complex 4. Complex 4, also known as cytochrome C oxidase, where it contains the cytochrome A and also cytochrome A3 here. And also in this complex, is not shown in the diagram, is 2 copper atoms, Cu copper atoms, also in this protein complex, but it's not shown in the diagram. Okay, so let's see what happened in complex 1. You see here in this diagram, what molecule that passed the electrons to complex 1? The answer is NADH. Okay, NADH. So NADH that we got from the lineation, from the glycolysis, from the Krebs cycle, it will pass its electrons to complex number 1. Okay, so electron pass through complex 1. NADH transfer the electrons to the first molecule, which is a flavoprotein with prostatic group. FMN stand for flavin mononucleotide. So you see in this diagram here, this is the prostatic group FMN. This is the prostatic group, the one that accepting the electrons from the NADH. And once the FNM receive or accept the electron, it will become reduced. Okay, it will become reduced. And what happened to the NADH? NADH, once it passes its electrons to FMN, it will become oxidized. Oxidized to what? Oxidized to NAD+. So this NAD+, okay, once it being oxidized, so it will then return to the Krebs cycle. It will then return to the link reactions and to accept another electrons from the process. And once it accepts another electrons, it will reduce to NADH, and then again go to the inner membrane of mitochondria, go to the electron transport chain, and again pass the electrons to the FMN. Okay, so the cycle will repeat continuously. And then what happened to the FMN? The FMN, once it receives the electron, when it accepts the electrons, it becomes reduced. After it becomes reduced, then it will then pass the electrons to the iron sulfur center. it will pass the electron to iron sulfur center. Once it pass the electrons to iron sulfur center, it will become oxidized. And the iron sulfur center will then become reduced after it accept the electrons. Okay, so that's what will happen lah. After that, we go to the next one. Okay, once we have the iron sulfur center here, accepting the electrons, then it pass to this one. It pass the electrons to the coenzyme Q. Co-Q is written here. Co-Q. Coenzyme Q, also known as ubiquinone. Okay, ubiquinone, or the full name is coenzyme Q, or can be also written as co-Q here. Co-Q. Capital Q. Okay. So what is coenzyme Q? Is a small mobile. Mobile means it can go here and there, can move around, and then it is a hydrophobic electron carrier. It's a hydrophobic electron carrier. This is the only non-protein ETC member. Okay, the rest all are proteins. Only the coenzyme Q is the non-protein electron carrier. Okay. So coenzyme Q will accept the electron, it will become reduced, and the iron sulfur center just now, after it passed the electrons to the coenzyme Q, it will become oxidized back. Okay, and then what happened to coenzyme Q? The coenzyme Q will then pass the electrons to the next neighbor. Who is the next neighbor? You can see the arrow here. The next neighbor is cytochrome B in complex 3. Okay. Okay, next, coenzyme Q will pass the electrons to cytochrome B, and then cytochrome B accept, become reduced, and then cytochrome B pass the electron to the iron sulfur center, and it become oxidized. Iron sulfur center accept the electrons and become reduced, and then iron sulfur center pass the electrons to cytochrome C1 and become oxidized. Cytochrome C1 accepting the electrons become reduced, and then pass the electrons to the next neighbor, which is cytochrome C. So this is what happens in complex number 3, which comprise of cytochrome B, next iron sulfur center, and next cytochrome C1. Okay, and then next cytochrome C1, cytochrome C here, cytochrome C1 pass the electrons to cytochrome C. So cytochrome C, just like coenzyme Q, it is also mobile. Mobile means this electron carrier, they can go here and there, they can move around. Okay. Then cytochrome C then shuttle or transfer the electrons to the next neighbor in complex number 4 to the cytochrome A. Okay, and then cytochrome A pass the electrons to cytochrome A3. Okay, last one, what happened to the electrons that is being accepted by cytochrome 3? Finally, cytochrome A3 will pass the electrons to the final electron acceptor, the oxygen. Okay, so oxygen is the most electronegative of all the electron carrier here in this electron transport chain. And oxygen, besides accepting electrons from the cytochrome A3, oxygen also will accept 2 hydrogen atoms from the surrounding, and the product will be water. So that's why in aerobic respiration, the product, water is one of the product. Okay, because oxygen here accept the electrons together with the protons and will form the water. So that's all for just now. I have explained to you guys how NADH pass the electrons to FMN of complex 1, and then goes to cytochrome coenzyme Q, and then goes to complex 3, and then goes to the cytochrome C, and then goes to complex 4. And in complex 4, we have the cytochrome A3 pass the electrons to oxygen and become the water. Okay, the next electron carrier also produced during Krebs cycle is FADH2, isn't it? So FADH2 also will carry the electrons and pass the electrons to the electron transport chain. But it passed the electron different to the different complex. NADH2 will pass electrons to complex 1, but FADH2 will pass the electrons to the lower energy level, which is at complex number 2. Okay. So another source of electrons is for the electron transport chain is FADH2. So you can see here, the FADH2 pass the electrons to FAD and it passed to the ion sulfur center. And then the ion sulfur center, once it got the electron, it will then pass to back to the coenzyme Q, and then coenzyme Q pass to the complex tree to the cytochrome B here. So you just see the arrow here. The arrow here already show us by sequence, the electrons transport by sequence. Okay, so that's why NADH, remember NADH pass electron at complex 1, FADH2 pass electrons at a lower energy level at complex 2. That's why at the end of the day, NADH passing electrons to complex number 1 will at the end yield more ATP compared to the electrons contributed by the FADH2, because FADH2 pass electrons at the lower energy level. At the end of the day, it will produce much lower ATP compared to the ATP contributed by the electrons from the NADH. Okay, because FADH2 pass the electrons at lower energy level. NADH at the end of the day will produce 3 ATP. One NADH will at the end of the day will produce contribute to the production of 3 ATP, whereas for the FADH2 at the lower energy level, they pass electrons at the lower energy level, it will contribute to the production of 2 ATP only. Okay, so one NADH contribute production of 3 ATP, FADH2 contribute production of 2 ATP. That's the difference. That will give out different during when we want to calculate the number of ATP produced at the end. Okay, so now it's questions time. I want you guys to try to answer these questions before we proceed. Okay, first question. Oxidative phosphorylation. ATP found using energy derived from the redox reaction of an
Electron transport chain. Electron transport chain.
electron transport chain. Okay. Alright, correct. Next. This involves electron transport chain coupled with what process?
Chemiosmosis.
Chem
Osmosis. Chemiosmosis.
Yeah, chemiosmosis. Chemiosmosis. Okay. So oxidative phosphorylation involves electron transport chain coupled with chemiosmosis. Okay, next. Most of the ATP comes from the energy in the electrons carried by
NADH.
NADH.
NADH, correct. And also
FADH2. FADH2. DH.
Yes, FADH2, correct. To the electron transport chain. Okay, then the energy in these electrons is used in the electron transport system to power ATP
Synthesis.
Synthesis. All right, correct. Okay, what is electron transport chain? Electron transport chain is a collection of molecules served as electron carrier, correct. Embedded in the inner membrane, correct. Where are the rest?
Membrane.
Here, Michelle's inner membrane of the mitochondria, correct? Okay, inner membrane is folded into
cristae.
Folded into cristae. Okay, correct, cristae. Next, most components of the chain are proteins which exist in multi-protein complexes, correct? Tightly bound to these proteins are
Prosthetic.
Prosthetic groups, correct? Non-protein components which accept and donate electrons. Okay, during electron transport along the chain, electron carriers alternate between reduced acid picks up electron and
oxidized
Oxidize, correct. In aerobic respiration, the final electron acceptor of the chain is
Oxygen.
oxygen, correct. Okay, next one. As electrons pass through the chain, a series of exergonic reactions release small amounts of
Energy.
Energy.
energy, right? Okay, let's see this one. Electron pass through complex 1. NADH transfer the electrons to the first molecule, a flavoprotein with prosthetic group. Prosthetic group what?
FMN.
F.
F. Flavin mono.
Yes, flavin mononucleotide FNN, correct. Okay, NADH oxidized to NAD plus, FMN reduced to FMNH2, later become oxidized as passes electrons to N.
FES.
Iron sulfur. It's iron
Iron
sulfur.
sulfur, iron sulfur center, correct. Okay, next. FES passes electrons to
Coenzyme Q.
Q.
coenzyme Q, correct. The only
None.
non-protein ETC member, correct. Okay, next. Coenzyme Q passes electrons to molecule in complex.
2. 3.
No. Complex
3.
3. Okay, complex 3. Cytochrome B pass electrons to iron sulfur center next to
Cytochrome C1.
cytochrome C1. Okay, correct. Okay, next cytochrome C1 pass to cytochrome C, which is the
Mobile.
Mobile.
mobile, correct. Shuttle that pass electrons from complex 3 to complex
4.
4.
complex 4. Okay, correct. Next one, the last cytochrome A3 pass electrons to
Oxygen.
oxygen. Correct. Good. Okay, each oxygen atom also picks up a pair of
Proton. Proton.
Proton.
proton from the proton from the aqueous solution forming
H2O.
Water.
water. Okay, correct. Okay, another source of electrons is the FADH2. FADH2 adds electrons to the EDC at complex
2.
2. Yes, in this diagram you can see at complex 2. Okay, correct. Okay, that's all for electron transport chain. Thank you. Okay, now very good. You can answer my questions. Okay, now we will proceed our lecture for next one is chemiosmosis. Okay, chemiosmosis, which is the proton motive force. So just now we have learned about the ETC, but just now when we learned about the electron transport chain, we learned about the electron transport chain just now. The